Okay. Okay. Hello, everyone. Good afternoon. My name is Fatima, and I'm a final year PhD student at TMU. I'm in the molecular science department. And today I'll be talking about my project here on a protein called polycystin 1 and how it's implicated in a disease called ADPKD. So what is ADPKD is one of the two types of polycystic kidney disease in which you see the progressive development of cyst on the kidneys. And ADPKD is the dominant type, which is also relatively more common between the two. And it's generally an adult onset disease with a prevalence rate of 1:400 to 1:1000. It's caused by mutations in mainly these two genes, PKD1 and PKD2, with which both of which encodes a protein called polycystin 1 and polycystin 2, respectively. And majority of my research work will be focused on polycystin 1 protein because it has the most prevalent mutations found in ADPKD. Now, together, both of these mutations, they result in are the fourth leading cause of end-stage renal disease, which requires the patient to either undergo renal transplantation or be on a lifelong dialysis. So currently, there is no cure for it, but there are treatments available that provide symptomatic relief here. As such, there is no standard therapy. There is one which is tolvaptan, which is a vasopressin antagonist. It helps in balancing out the sodium levels in the renal epithelia and thereby reducing the enlargement of the cyst. But it comes with potential, liver side effects. So it's still not a standard care. And in order to understand the polycystin proteins, it is very important to look at this organelle called primary cilia. Now, primary cilia, unlike the motile cilia, it's not motile, but it's the main signaling hub. It's like the antenna of the cell. So it is where the major cellular signaling messages get transmitted from. And if you look at the nephron of the kidneys, inside the renal epithelia of this nephron, these hair-like projections, if you see the magnified version of that hair-like projection is what the primary cilia here is. And it has a bunch of proteins going anterograde and retrograde trafficking along the length of the cilia, of which polycystin 1 and 2 are two of those proteins that that move along the length of the cilia, and thereby they regulate a lot of cellular signaling pathways. And if you look at the structure of PC1 and PC2, in particular, it's important to look at the C-terminal region here, in which both of these proteins they form a coil-coiled, they're interlinked. And through GPS cleavage, these fragments, they get cleaved and then they travel to different organelles of the cell, but also part of this fragment goes to the primary cilia. And thereby it regulates not only the messages that go for a variety of cellular proliferation signals, but also because PC2 is implicated in calcium ion channel signaling. So it thereby ensures the fluid balance within the renal epithelia. Also, the major mutations found in ADPKD are clustered in this region. So this will be my focus for the rest of my presentations, where we look at what are the interactions, in particular at the C-terminal tail region. So as I mentioned, the C-terminal tail region gets cleaved into three different fragments, or majorly three different fragments, one of which goes to the primary cilia, but the others go to endoplasmic reticulum, mitochondria, but also to the nucleus, where they activate these downstream signaling pathways that then ensure that there is the healthy proliferation signaling continues for the healthy division of the cells. Now, within the field of, within this ADPKD, there are a bunch of pathways, just as you would see. A lot of these are overlapping with the ones that are found in cancer as well. And because they are so interconnected, any single therapy that targets one particular pathway generally is not able to, arrest the growth of this cyst, because there are compensatory mechanisms in the cells that override the effects of these drugs, and they're able to still continue. The cystogenesis process goes on, and thereby the disease continues to progress. And this is one of the reasons why it is so difficult to find a single therapy to this state for ADPKD. So going forward, our rationale would be to look at what are the bunch of proteins that interact at the C-terminal tail region for both polycystin 1 and polycystin 2. And C-terminal region, because this is the major region where major mutations, as I mentioned earlier, they occur. So in order to assess the interactions of these proteins at PC1 and at the C-terminal tail region, we employ this technique called BioID, which is where you take your bait, which is your protein of interest. In this case, it would be PC1 CTT, and then you tag it with an enzyme called BARE. Now, BARE has an affinity that in the presence of biotin, when you externally supplement the media with biotin, in the presence of biotin, the BARE gets activated, and then it labels all the bunch of proteins that are found within the vicinity of your protein of interest. And then you pull down this complex and you shoot it through mass spec, and it gives you the list of proteins that are the strong interactors with your protein of interest. And once you assess that, you get something like this, which is where you see in different compartments of the cells, they get enriched with a variety of these proteins. For our focus, and particularly, we found this block-Borg complex that was a standout, which is involved in the vesicle trafficking. And this protein complex was highly enriched in polycystin-1 interactome, but it was missing in polycystin-2, but also missing in the mutant form of polycystin-1 that we did the BioID on. So this complex then we thought would be very critical in assessing the interaction of how it affects and modulates the regulation of polycystin-1. So this is just a dot plot that shows that how greatly the both the block and Borg complex is enriched in the ciliated polycystin-1, and ciliated, by ciliated, I mean it's a serum starved condition where you would normally in the nutrient deprivation condition, you would see a lot of autophagy factors also, they get upregulated, which is what you would find in an ADPKD like microenvironment as well. But in the regular polycystin-1 non-ciliated condition, they're still present, but they're not as greatly enriched, but they're completely missing in the polycystin-2. Also not shown here is the mutant data, in which you don't see an enrichment of any of these block complex proteins. So we wanted to then confirm what is actually the role of this block-Borg complex proteins, and its relation to PC1. So just a brief about what this complex is. This complex consists of 13 different proteins, of which 3 are the shared interactors between these two complexes. And block 1 is required for the biogenesis of lysosomes. Lysosomes are this recycling bin of the cell, where all of these different, cellular, debris and everything is kind of sent to for degradation. And Borg assist in recruiting different proteins as a cargo proteins on microtubule for the microtubule dependent, trafficking. And so for the BioID, we saw that the block 1 and block complex has a strong association with the PC1, but we wanted to see how that shows up if it is a true interactor, because BioID is not like, it doesn't give you a very direct indication of whether it's a direct or indirect indicator. So we wanted to then see if it is physically interacting or not. And for that, we did the co-immunoprecipitation. So this is a pull down data here, where in the presence of PC1, we were able to pull down 3 of the main subunits of block and Borg complex units. And wherever there was PC1 absent, the block complexes were also not, they also were not able to be pulled down. And then we further, to kind of revalidate that, we also did the proximity ligation assay here, which is nothing but a more qualitative way of assessing if SNAP-in or any of these block complexes are the true physical interactors of PC1 or not. And as you can see by the signal intensity as well, these proteins are very strongly interacting with PC1. So then we wanted to see what is the effect, because in the context of primary cilia, how the knockdown of block complexes is affecting polycystin-1 protein. So in the healthy cells expressing primary cilia, or both the polycystin-1 as well as the block complex, they localize at the base of primary cilia. So this red hair-like structure is the primary cilia, and what you see is the green dot is each of these block complexes, and here is PC1. So all of that, they go at the base of the primary cilia and they localize there, and that's how they regulate the cellular signaling along the cilia. So once we wanted to assess if you knock down, and also we did, I think, one of the different cell lines, we also knocked out to see what is the effect when in the absence of block complex subunits, if PC1 is able to still localize to primary cilia base or not. And what we found was sort of what we expected also, is that although the total protein levels of polycystin 1 remain unaffected, its localization to the primary cilia definitely in this case gets disrupted. So you see the disappearance of the primary cilia, PC1, from the base of primary cilia here. And so then we wanted to see how, because block complexes is involved in lysosomal biogenesis as well. So it is known that in PKD field, the knockdown or the sort of a mutated version of PKD1 results in the aggregation of lysosomes, because there is some sort of a correlation with that. And there is a strong correlation with aggregation of the lysosomes observed in PKD1. So it is known. So we wanted to see if it has a similar effect, if it is the knockdown of block complex is able to mimic the effects of PKD1 or not. And what we did find was that it does have a near similar level of lysosomal aggregation, or more like perinuclear clustering, as you can observe from here. And so then, and similar to the so this was done using the lysotracker assay. And then we wanted to further, reassess that using a DQBSA assay, which is also measuring the presence or labels all the acidic, organelles of the cells. And in this case, again, the block complexes were able to, in the healthy condition, were able to localize alongside the PC1 in the presence of these lysosomal compartments as well. So with all of this data now indicating towards the possible role of block complexes in with PC1 in ciliary localization as well as in lysosomal phenotype, the final stage for us was to assess how much of the effect of this block complex knockdown has on cyst formation. And it is already known that the PKD1 and PKD2 knockdown increases the cyst, results in cyst formation in the in vivo as well as in vitro models. And so we wanted to see if the knockdown of these block subunits has a near similar effect like PKD1 knockdown as well. So we did this 3D organoid, these are this IMCD3, which is a mice cell line cells, which are lentiviral transfected. So we carried out a lentiviral mediated knockdown of these individual block subunits. And we seeded the cells in Metrigel. And in the absence of any of the growth factors, their growth, their proliferation rate, as well as their enlargement, was observed for the span of 4 to 6 days. And what we found here was that not only the number of cysts were more in all of these four, data sets, but also compared to the scramble, which is just the negative control, but also the NO6, which is known that the effect of NO6 upon knockdown results in, arresting the growth of the cyst. So this acts as like a negative control here. So we found that the number of cysts as well as the enlargement in the cyst area, which is nothing but this uptake of the renal epithelial fluid, the aggressive uptake of the renal epithelial fluid into the cyst cells, result in higher enlargement of the cyst. So both size as well as the area were greatly increased in the block knockdown samples, which were near similar, still not as similar to the PKD1, but almost definitely much higher than what you would see in the negative control. And the final leg was here for us to see that the regular healthy phenotype of these cells in the absence of any cyst forming mutation is that they form tubules. So we wanted to see that firstly in this case, these are the block knockdown cells, which I showed you in the previous slide, that they form cyst under Metrigel, but once you are overexpressing this with PKD1 wild type, so basically we try to rescue the blocks knockdown phenotype with the PKD1. So we supplemented them with the healthy PC1 CTT, and we expected whether they were still able to form cyst or whether it rescues its healthy phenotype. And to our surprise, like this was a really interesting finding here that they reverted back to the tubular phenotype here, which is what you would expect in the healthy cells, which does not have any mutation. So this again tells us that the supplementation of the full length PKD1 in the background of the block knockdown cells is able to help the cells revert back to their healthy phenotype here. And finally, since we know that this 13 different subunits of block Bohr complex are related with PC1 in direct, with a direct binding with PC1, we wanted to further assess which is the single subunit here that is a clear standout, which is holding the whole complex together. And so that could in future be explored for a more, therapeutic targeting. And after like a bunch of troubleshooting, which resulted in no clear conclusive data, we finally came across this assay called split luciferase, and we employed this. So this is where we cloned or designed each of these block complex constructs with that N-terminal N having the small bit here, which is similar to the probe that you see in the PLA here. And then the large bit, which is the second construct, which has our main protein of interest, which is PC1 CTT. So you're assessing the binding ability and the strength of binding here. And once you so these cells were like co-transfected in HEC293 and transiently transfected, and then upon addition of the luciferase substrate, if the binding is really strong, then it will generate a corresponding higher signal intensity, which is measured in terms of bioluminescence. So here the heat map shows that the S2 has the highest signal intensity, followed by S1. But if we are to look at, in future, analyzing any of these subunit domains for its major interactions with PC1 and therefore, its potential for therapeutic targeting, then we could largely look at S2 and S1 because they've so far showed, at least in this assay, to be very strongly, have a binding capacity with PC1 compared to all other block subunits. And so in summary, the BioID data set here identified some very important shared interactors, but in particularly, it was the block Bohr complex that stood out here. And this complex was found to have a very direct interaction with PC1, which affects its ciliary as well as its lysosomal trafficking. And once you knock down this block complexes, it's affecting not only the localization of PC1 in the cilia, but it also results in enhanced cyst formation. And secondly, it is also similar to mimicking the lysosomal aggregation phenotype that you would see in the PKD1 mutant condition. Finally, the overexpression of this PC1 CTT is able to rescue these block complex knockdown effects and result in tubulation. And in particular, these two subunits, they appear to be very strongly interacting with PC1 CTT, offering the potential for future therapeutic targets. And finally, I would like to acknowledge my PI and team members here, our collaborators, committee members, and the various funding agencies. With this, thank you, and I'm open to taking questions now.
Awesome. Thank you so much, Fatima. That was really interesting. Feel free to type your questions in the chat to Fatima or raise your hand and we can unmute you. I do have a couple questions for you though. Your PC1 protein, do you know if in the other interacting proteins, if it interacts with any other coiled like proteins, or it does, I think it was your block 1 also that interacts paranuclear lysosomes. Do you know if it interacts with any nuclear membrane proteins also to like either leak something out or?
So far, I think there are a bunch of proteins interactors here, particularly in this data set stand. So you would be looking at some of the ER tethers proteins. I've not included the membrane trafficking proteins here, but the N-terminal region of the PC1 because it has a very large N-terminal extracellular region, it kind of loops back, and it
Right.
also interacts with a bunch of proteins. But the major interaction of this is with PC2 only.
Right.
But
Okay. Okay, cool. Thank you. And then we have a question in the chat. Did you notice in your PLAs if there were any changes in subcellular distribution on observed interactions with different interactors or mutants?
with, sorry, different interactors.
Oh, yeah, or mutants. Yeah.
With the PLA, so yeah, so with the PLA, I've not included the data with the mutants here, but we did do it in order to rule out if it has any, potential. Sorry, one second. Yeah, so I we did include. We just looked at the majority of the block complex, majorly these two subunits, but we also tried to look at the. So there is the Q4 mutant for PC1-CTT, which we've also used for BioID, in which the block complex is not interacting, and we've used that for the PL as well, but we did not find any signal intensity. So for that, the signal intensity of it was near similar to what you would see in the negative control. So it's completely not interacting, indicating that the mutant PC1 tail is failing to form any binding with any of these block subunits. If that answers your question.
Yeah. Oh, we'll find out. Okay. I think it does. Perfect. Any other questions for Fatima? Okay, if not, thank you again so much. We really appreciate your presentation and taking the time, and good luck on your defense. You're going to be
Thank you so
amazing.
much.
Perfect. All right, we'll switch gears over to our postdoc presentation. Saby, feel free to start sharing your screen.
Hi. Yeah, I don't know why I typed that. I should have just asked her straight up. But yeah, great talk. It was really cool listening to Fatima's talk. Yeah, let me just share my screen real quick. So hopefully you can see everything. Yeah, it's nice to give a talk for CSMB. That was actually my first meeting ever. Actually, my former supervisor's right there. He's looking very fancy there. So it's a core memory for me. It's nice to give a talk with other Canadian scientists. I did my undergrad honors thesis on dictyostelium in Rob's lab. And then I did my PhD at Dalhousie University on promyelitic leukemia, where we had a bunch of different fun models, including the zebrafish. And now I'm in Boston studying solid tumors, and I'm trying to understand a little bit more about them and really how they develop chemotherapy resistance. So that's what I'll be talking about today. And hopefully I'll get you excited about these weird looking cells that pathologists have always noticed. They're called these polyploid giant cancer cells. And so I'll talk about that today. And I'm a member of the David Langenau lab. So I've been working really closely with this postdoc in our lab named Tiffany, and this is kind of a joint effort on this project. So yeah, aside from the dramatic name, I'll be talking about these polyploid giant cancer cells. And really our lab is focused on rhabdomyosarcoma, and it's a skeletal muscle cancer. It's a solid tumor, and it comes in three flavors. There's three subtypes known as, one is the fusion negative, which I'll primarily focus on. That's commonly caused by mutations to the RAS pathway. But then you also have this fusion positive type of rhabdomyosarcoma, and that one is primarily driven by chromosomal translocations, leading to fusion proteins that really drive oncogenic transformation, primarily PAX37, FOXO1. And then you have this other subtype, far more rare, but very aggressive therapy-resistant form known as the mutant MyoD subtype. And so our lab's interested in all of these, but fusion negative is kind of the focus today. And typically for therapy, patients receive radiation, chemotherapy, and surgery. And this works most of the time. About 70% of the time, you see really great responses. But in 30% of patients, not so much. And really, the survival rates are abysmal when you do have relapse and chemotherapy resistance. So our lab's really interested in figuring out how this happens. And we think one of the keys to this puzzle behind how these cells are obtaining therapy resistance comes down to these weird looking cells often called therapy effect. So after treatment with chemotherapy, you'll notice these large cells with multiple genomes, polyploid, and they're giant in size. And typically, we refer to them as just therapy effect, but people are getting more and more interested in these cells. They're often missed in single-cell RNA-seq, so there hasn't been many easy ways to study these cells. And so I'm hoping to convince you that they are important and they're doing something interesting here. And really, their role seems to be related to the cell state heterogeneity in rhabdomyosarcoma. And just as a quick little overview, cell state heterogeneity is really referring to these transcriptional clones that are existing within the same tumor population. So genetically, they're the same, but they have different epigenetic programs, so they behave slightly different. And these behaviors actually have a huge influence on how these cells respond to therapy and behave in terms of the tumor response and the tumor growth. And so our lab, along with 3 other labs around the world, work together to perform single-cell RNA-seq and really figure out what's going on in rhabdomyosarcoma in terms of these cell states. And so quite a few different models were used, including PDXs, patient-derived xenografts, and actual primary patient samples, and then cell line models. And we kind of combined all these models. And my former mentor in the lab, Claudia, she really established this single-cell map showing that you have these 3 dominant cell states, one referred to as this proliferative population, which is making up the bulk of the tumor. And then you have this differentiated population that actually looks a lot more like mature skeletal muscle. It's not differentiated muscle in the sense that it doesn't look like muscle fibers. There's no syncytia. It doesn't necessarily behave like normal differentiated muscle, but it's exhibiting features transcriptionally that resemble differentiated skeletal muscle. And then you have this third population that our lab has spent most of our time thinking about. It's this progenitor population, and they seem to be expressing unique markers like CD44, CD90, and any cancer stem cell people here will realize like that is like a very standard marker for cancer stem cells, and that's really what these cells are, these progenitor cells. They look like mesenchymal cells during development, and they have features that make them quite interesting in the context of therapy response. Mainly, the most important feature is that they're therapy resistant, and they seem to be activated by stress. So you can imagine these cells are really the ones driving resistance and relapse during rhabdomyosarcoma progression. And an important feature of these cells is the fact that if you take these cells and transplant them, they're able to reform the tumor. So they behave like cancer stem cells. And so in the last 2-3 years, now that we have markers for studying these cancer stem cells, these progenitor cells, my project's kind of been related towards describing them. And here was a really nice paper by Sarah Dinelli. Claudia was also involved from our lab, and she basically showed that essentially when you take patient tumors before and after therapy, you can see a shift in the progenitor signature. So that same gene signature that I referred to for the cancer stem cells seems to be enriched after therapy. Really, again, supporting or highlighting the idea that these cancer stem cells are an important component to how rhabdomyosarcomas obtain therapy resistance. And actually, this was a fun experiment. If you stain these different cell states, the differentiated, proliferative, or cancer stem cells, you can see these beautiful little niches forming within the population, in particular with the differentiated muscle as well as these cancer stem cells. They exist within these little niches next to ground cells that are the most plastic. They don't really have features of any specific type of cell state, but you can see that there's very much niches that form even on a 2D plate between these different cell states. And so we were quite interested in understanding this signaling field effect that was going on potentially. And so to do this, Tiffany in our lab, who's very excited in this photo, she came up with a very cool model or a reporter line to study this. And so essentially, she took EGFR, which is very enriched in these cancer stem cells, and she generated this very cute reporter that turns on dendra, which is just a fluorescent protein, green fluorescent protein, which is photoconvertible, hence why we used it here. And what she did was she wanted to see if we could actually mark the cancer stem cells to study their behavior and how they respond to therapy. And so it works really well. Essentially, throughout this talk, I'll point at green cells, and hopefully you'll believe me that those are those cancer stem cells. And so that's what we're trying to mark here. And we did a lot of characterization to really prove that that was the case. So here you can see that these dendra positive cells are also enriched for CD44, CD90, which were those other markers for cancer stem cells that I was referring to. And then if you actually take these cells into tumor sphere assays, which is, again, that classic assay to prove that these are the cells that are reforming the tumor, we can see that it's much more increased. I haven't shown it here, but we've done all these experiments to really validate that this reporter is doing what it's supposed to do, which is marking those cancer stem cells. And the really exciting part is you could do very cool live cell imaging of the cancer stem cells and their behavior. So you could watch as they do symmetric stem division in terms of making more of themselves, right? So making more of those cancer stem cells. You could see symmetric differentiation where they're becoming two differentiated cells. And then finally, you can also look at asymmetric division as well. So one retains that cancer stem cell signal while the other cell becomes differentiated. So you can actually live track this and see how you can see how stress might affect the different differentiation or division conditions for cancer stem cells. And this was pretty exciting, and it lets us now answer that question of what features or what dynamics are really going on with these cancer stem cells. Oops, I don't know if, there we go. Whoops, too far. And so the next question, of course, is if you hit them with therapy, what happens? And so when you do a single dose of VAC chemotherapy, this is really exciting. If you wait 2-3 weeks, you'll actually notice that the cells become much more green within the population. Again, fitting our hypothesis that the cancer stem cells are really important for therapy response. But another interesting feature is the fact that these cells seem to be the only ones really surviving initially. And then as you look, you can start to see these clusters, again, those niches. One full of these really messed up looking cells, and other cells that look somewhat normal, monoploid, healthy cancer stem cells, like green positive cells. And so you have a mix of both within this population. But essentially, the tumor before treatment looks nothing like the tumor after treatment. So once you have a newly confluent plate of these previously treated cells, it's almost like these cells have memory of that initial dose of therapy, even 2-3 weeks later. So you could see that these cells are really transformed over time. And we wanted to understand how that was happening. And one thing we noticed with our live cell tracking was the formation of these guys, these weird looking cells with multiple nuclei that try to split, but can't complete abscission. So they come back together, just kind of slap back together. And now you have this monster looking cell with multiple nuclei, a lot of chromosome fragmentation, and really something that looks very genetically unstable. And it is. And we thought for a long time that, because we'd always see these cells in our samples after therapy, and we always thought these were dying cells, but then we did time courses to actually track these cells, and it was really interesting. We started to notice that these cells would actually move along, and almost like Tesla Cybertrucks, you can't miss them. They're just moving, and they're causing a lot of these issues around the cells, in the sense that a lot of the cells around these large cells, these polyploid giant cancer cells, or PGCCs we call them, they seem to be becoming more and more green. And this got us excited because it was suggesting that maybe these cells were actually transforming the cells around them. Let me just skip ahead. And then when you quantify this, you can see actually these PGCCs pop up very early on during therapy response. So actually within the first 4 days, you can see a huge increase in these giant cancer cells. And they persist, even though the percentage of them decreases as these other non-PGCCs start dividing later in therapy, after therapy. These PGCCs seem to dominate that initial therapy response. So these giant cancer cells, they're present, they're doing something, and it looks like they're not just dying cells. They seem to be migratory and very active, which was unexpected. And then we did some reading. We were curious about these cells, and it seems like it's quite common. You can actually see these cells in glioblastoma, as well as prostate cancer and all sorts of different tumor types. The more digging we did, the more and more types we found. And honestly, it looks like it's just a new theme to a really messed up cell that's always been present. And then we did our own work looking at mouse PDXs after engraftment and treatment, and we noticed that we do see these giant cancer cells present, which is what you see in the blue arrows. And then also in patient samples. So that right there is a tumor from a 6-year-old boy before and after therapy, and you can see the presence of these giant cancer cells. And so we're really interested in understanding these cells and what's going on. And so with that idea that these cells were maybe transforming the cells around them, we did this fun experiment. We had a summer student in our lab, super smart. She's from France. She did this cool experiment where she took cells, which were labeled with BFP, and she irradiated them. And then what she did was, if the idea was that if you could irradiate them and they primarily form giant cancer cells, which they did. Actually, 90% of the cells, once you irradiate them at this higher dose of 6 grays, most of them were giant cancer cells. And then you mix them in at a smaller ratio with naive cells, those reporter line cells, the cancer stem cell reporters. What we were hoping to see is that potentially you can study how these PGCCs in blue were affecting the cells around them. And then we would compare it to our untreated group that should divide and behave as normal. And then what you're seeing here on the right is what we saw, which is pretty exciting. You can see a really striking increase in the amount of dendro signal after you throw in these PGCCs in the mix compared to the untreated cells, which are actively dividing. One thing that's also interesting is that you can notice that if you label phosphohistone 3, you'll notice that the cells seem to be dividing farther and farther away in the case of the PGCCs, but for the normal cells that are untreated, you don't see this behavior. So what we think might be going on is that potentially you're not only increasing the number of cancer stem cells around these PGCCs through whatever mechanism, but you're also seeing a quiescence zone. The cells around these giant cancer cells don't want to divide. And you can imagine during chemotherapy, when division is the main mechanism for killing these cells, telling a cell not to divide is a scary thought, right? These cells seem to be providing some type of cue to these other cells, telling them not to divide and to become cancer stem cells instead, these dedifferentiated cells. And so we had this working model. The idea was that you have a progenitor cell state, which normally won't divide and will survive therapy. But then the cells that do try to divide, some of them will become these PGCCs after DNA damage. And then when they become PGCCs, they seem to be promoting the formation of more of these progenitor cells, really accelerating therapy resistance and relapse in fusion-negative rhabdo, and potentially in other tumor types as well. And so even though we think this is what's going on, we don't really understand how this is happening. And so our thought was that maybe it's a cell-to-cell communication thing. And so we had a couple fun ideas to study this. We didn't know if cell contact was really important. So these were the first 2 experiments to really prove that or see if cell-to-cell contact was necessary. Actually, this is what I did in Rob's lab back when I was an undergrad. We were doing a lot of paracrine signaling stuff. So I was so excited to get back into paracrine signaling. And so what we did here was a similar experiment to before, where you would take the naive cells, but in this case, instead of directly mixing in the PGCCs, what we were doing was we would grow these cells on inserts, treat them with therapy, and then put them on top of naive cells. And the insert basically has a filter that's allowing media exchange, but the cells are still separated. And so this was one approach. This was a co-culture approach, very classic approach. And then the second approach was taking the conditioned media from therapy-treated cells and putting it on naive cells. So really 2 ways to test the same thing with slight differences, right? One would be looking more at cytokines, one would be looking more at like exosomes and those features. But essentially, we would then do confocal and flow cytometry to see if there's any changes to cancer stem cells. And we did this fun experiment, and the result was really striking. What we noticed was that if you do co-culture or conditioned media, you see a very similar increase in cancer stem cells, which are marked by green again here in our reporter line. And regardless of VAC chemotherapy, which is just this combination of chemotherapy, I should have mentioned that earlier, or radiation, you see a very similar effect. Essentially, you're seeing a very striking increase. Even in the absence of therapy, whatever is being secreted into the media is sufficient to actually transform these cells. And so we were, of course, curious about what's actually going on in the media that's causing this. So similar experiment, we took conditioned media from treated cells, and we put it on naive cells, and then we did RNA sequencing after 5 days. And what we saw was very striking. We saw that interferon was really upregulated, which was a bit of a surprise, but then we compared it to our patient data sets before and after therapy, and we also saw interferon upregulated there. So it seems to be a pathway that's quite enriched, and you would expect interferon to be up there after therapy because we figured, DNA damage, this is going to trigger some type of stress pathway. We just didn't expect it for tumors that were usually considered cold tumors to produce such large amounts of interferon. So we were curious, is interferon really playing a role? Or is it something else? It's a long list of genes or pathways involved. It could be any of them, but interferon was our top hit. You also will see like EMT, which is really a marker for that mesenchymal signaling that I mentioned, and that's usually been associated with those progenitor cells. So we are seeing a cancer stem cell signature alongside that interferon signature there as well. And so we did proteomics first, and then we validated it. So we took the conditioned media into proteomics, and we noticed that interferon was in there. And then we validated it by ELISAs, where we actually measured the amount of interferon in the media, and we noticed it was a pretty striking amount. Usually, when you look for interferon, it's very low amounts of interferon, but even under basal conditions, you could detect interferon being secreted by these cells. So they seem to have already seem to be prone to secreting interferon. There's high levels of DNA damage naturally, but after therapy, which is what you're seeing here with orange showing VAC chemotherapy, and then irradiation in blue, you're seeing a really huge increase in terms of the amount of interferon being secreted into the media. And then if you actually just take the VAC conditioned media and throw it on cells, and you look at ISG15, which is a marker of interferon, you can see that's only pretty much in those VAC conditioned media treated cells. And then if you just take interferon and throw it on cells, you'll see the opposite, where you see, or not the opposite, but you'll see the other side of it, which is that dendra signal is increased. So just adding interferon seems to be sufficient to promote a cancer stem cell signature, as well as the ISG15 signature, which is what you would expect. And then we've now been we're working with some groups in St. Jude as well as Mass Gen to actually stain this ISG15, which is a good marker of interferon. And here you can see after therapy the presence of these giant cells, as well as a strong signal of ISG15, really highlighting that after therapy, interferon seems to be upregulated. And then this was kind of the fun experiment. I would say, like from my postdoc, this was a cool experiment, the one cool experiment, which was we did a FACS sorting experiment where we actually separated the different populations. The dendra reporter lets us now mark the cancer stem cells from these other cells. So what we did was I treated cells with therapy, and then I took conditioned media from dendro-negative cells, so either the differentiated or the proliferative cells. And then I took conditioned media from cells that were dendropositive, so cancer stem cells. And then I finally took conditioned media from large dendropositive cells, which were those PGCCs. And one thing that was really cool is if you take that conditioned media after isolating these cells, and you put it back on naive cells, what you'll notice is that these giant cancer cells seem to really cause the conditioned media to change, cause the greatest effect on the naive cells. So you see a really strong increase in ISG15 as well as dendra. The dendra-positive cancer stem cells that are monoploid seem to also have some effect, but the PGCCs seem to have the most striking effect in terms of transforming naive cells. And this was exciting. When we actually plated these cells after sorting, what we noticed was that these large dendropositive cells, those PGCCs, seem to secrete a ton of interferon. They love secreting interferon. They're very genetically unstable, and they're secreting interferon. So we started to think that maybe these cells are functioning as almost like beacons or lighthouses. They're actively signaling to these nearby cells with interferon and promoting a cancer stem cell enriched environment. And then one of my favorite pathways from grad school, this is a stress response pathway. Essentially, what I'm showing you here is CGAS sting, which is when you have stress and specifically DNA damage, there's an accumulation of cytosolic DNA. And that cytosolic DNA has been sensed by CGAS, which is a sensor within your cytoplasm. And once CGAS senses DNA, it activates the stress response pathway by secreting cyclic GAMP with this dinucleotide, cyclic dinucleotide, and then that activates STING. And then you end up the secretion of interferon and other factors associated with type 1 interferon signaling. And this is pretty exciting too. You can see here that when we did, actually, it was really striking when we did electron microscopy on these cells, these PGCCs, we noticed a ton of micronuclei. They were just littered with micronuclei. And a lot of those micronuclei, they look like they had ruptured nuclear envelopes. And then we kind of confirmed this by actually staining for CGAS. And you could see these beautiful condensates that are very specific only in the treated samples in these PGCCs. Where you see these nice little condensates. Essentially, CGAS will form these condensates around micronuclei to really promote the activation of STING and their own activation. They impede the entry of TREX1, which normally degrades these micronuclei. And then if you look at some other downstream markers like TPK1, phospho-TPK1, you can see it's very enriched specifically in these giant cancer cells, PGCCs, after you isolate them. And then we've also done now Westerns. Still, we're like still in the process of repeating this and doing this properly, but you can see a really enriched signal for EGFR, which is a cancer stem cell marker, a reduction in myogenin, which is a differentiation marker, and then STING and phospho-STING seem to be enriched in PGCCs after treatment, which does not seem to be the case as much for progenitor cells or dendronegative cells. And so the last kind of experiment I'll talk about is a very simple experiment. We wanted to see is if you knock down the different components of the pathway, so if you knock down CGAS or STING or ERF3 using siRNA, and then you collect a conditioned media, we still get an effect. And so that's why I'm showing here. So we did siRNA transfections where we transfected in siRNA for CGAS or STING, and then we treated with therapy 24 hours later. And we did that same conditioned media experiment. We collected the conditioned media afterwards. And what we were hoping to see is, do you still see an increase in dendra? Is it independent? Is it dependent on this pathway? And no surprise, what we saw was really striking in terms of the response. Essentially, you see that increase in dendra signal as you would expect with that conditioned media. But if you knock down CGAS or STING, which are involved in that sensing and stress pathway, you no longer get a response. Really highlighting the fact that CGAS and STING are quite important for the signaling pathway, which is a bit paradoxical. Typically, when you think of this pathway in the tumor environment, you think of it as a good thing. You want to promote immune infiltration and activation. So typically, CGAS STING is considered a good pathway for tumor suppression. But here, these cancer stem cells seem to be hijacking this pathway for their own signaling. So as a working model for what we think is going on in fusion-negative rhabdo is the fact that these progenitor cells are existing. They're surviving therapy. They're not dividing. They're quiescent cells. But then you also have the effect of dedifferentiation from PGCCs. So DNA damage hits these cells. They try to divide these proliferative cells. They become PGCCs, a portion of them. And then you have hyperactive CGAS STING signaling, leading to interferon that seems to be the key cytokine for transforming the nearby cells. So you're really promoting dedifferentiation through these PGCCs. So I guess the big takeaway is that we think these are like signaling beacons that are promoting a quiescent therapy-resistant environment. And these are my wonderful lab mates who have done a huge job on this project. Tiffany's right there in the red sweater, who's done a really amazing job helping with this project and really driving it forward with me. So yeah, thanks for listening to my talk. I really appreciate it.
Thanks so much, Saby. That was incredible, as usual. Do you have any questions for Saby? I do have a couple, but I'll open it up to the audience first. Okay. I'll let you guys think about it, but I do have a couple questions. The only thing I know about PGCCs in general is that they're usually highly resistant to hypoxic stress. Have you tested anything under hypoxic conditions or anything in either of your cell lines to look at the formation of PGCCs later on?
We haven't yet, but that's a good question. We in the literature, it's like pretty well established that if you have hypoxic zones within the tumor, you can start to see the formation of these PGCCs.
Right.
And so one thing that's kind of fun is if you look at like a patient tumor after therapy, you'll actually notice the regions with a ton of angiogenesis happening, which are presumably those hypoxic zones, also have a ton of these PGCCs. So there is a correlation there. Maybe they're forming from hypoxic stress. And typically we will see like 1 to 2% of them, even in the pretreatment sample, like in the growing tumor. So it's possible that they're also forming in those sort of stress conditions, not just in the context of therapy.
Right.
But yeah, we haven't directly tested it, but yeah, it's a fun idea. It would be definitely related to DNA damage and stress, so it would work. It would make sense. Yeah.
For sure. Yeah. Okay, cool. I see a hand up. Rob. So, question.
Great talk, Saby. So have you thought about potentially the upstream process where the multinucleated cells are reassociating? Like, could that provide an opportunity for therapy intervention if you can somehow prevent the formation of these PGCCs?
Yeah, it's a good question. We think
And
about that a
what's,
lot.
I
Like,
guess a question tied to that is what's causing the cells to reassociate?
yeah, exactly. That's a great question too. So, I don't know. I don't have a perfect answer for you, but we've done some sequencing of these like cells because one thing we've noticed is that if you take interferon actually and you put it on these cells, you're actually getting some PGCCs again. So we think there might be an autocrine effect going on, and we think initially you have DNA damage, sequestering signaling, and then autocrine feedback signaling is causing these and continued PGCC formation. We think it's related to escort. So some of the abscission proteins are associated with the escort pathway. They seem to be downregulated, specifically after exposure to back conditioned media. So we think potentially escort could be a thing, but we haven't directly tested it to prove what's happening. And then to your second question, which is therapeutically, do you go after those multinucleate cells? That's a good question. I don't know yet. We're trying to figure that out because in my mind, I don't think the cells are a bad thing, but their signaling is an issue because
Yeah.
them forming is really I don't think we can prevent that because it's probably loss of P53. They have
Okay.
genetic cues that let them form. But if we can block the signaling, then we're preventing the part that lets them reprogram the cells around them.
Right.
So I don't think even if you look at prognosis data, it's not necessarily a bad thing to have. This is actually a cool study. They were showing that if you have 90% or more PGCCs, treatment's great. They work great. But if you
Hmm.
have any less, like if you have a partial effect, it's actually worse prognosis. So it seems like if you get to that like intermediate where they form, but they're not all PGCCs, it's not
Hmm.
good for the tumor or sorry, the patient. But then if they are primarily PGCCs, therapy is working the way it should.
Okay.
Um, yeah, but yeah, we've been thinking about that a lot.
Great. Thank you so much. Any final questions for Sabi or Fatima?
I guess just one more question if I can follow up. So what's happening to the cells that are targets of the interferon signaling? So you're looking sort of how interferon is being generated in the PGCCs. What's happening outside? What's interferon? It's beta, I'm assuming that you're looking
Yeah,
at,
interferon
or
beta. Yeah.
so is it binding to like a cell surface receptor on the neighboring cells? What pathways do you think might be activated in these neighboring cells?
Yeah, so we did flow for like the main receptor for interferon
Hmm.
beta, and it's really enriched after therapy. So we do think
Really?
the receptor is also following the same track, but it seems to be primarily through STAT1 signaling.
Yeah.
So it feels down STAT1. I think Tiffany is also working on like potentially other cytokines that could maybe synergize and really enrich for that pathway. But if you knock down STAT1 from back condition media, that's sufficient to turn off the response. So you
Oh,
don't
really?
get,
Okay.
yeah. So actually, therapy
Cool.
wise, that would probably be a better target because STAT1 inhibitors are more available, or STAT
Hmm.
inhibitors in general. So we were thinking maybe you could synergize that and just go after the cells that are receiving the actual signaling, right? Those
Exactly.
secretors, yeah.
Perfect. Awesome work.
Yeah, thank you.
Awesome. Okay. Well, thank you so much again, Sabi and Fatima, for presenting. I really appreciate it. Our next seminar is going to be Tuesday, February 17th, so feel free to join for that. We have a couple of great speakers as well. But thank you so much again for coming, everyone, and feel free to reach out to anybody if you have any questions.